Hi everyone, this little video looks at Key Area 4 of Unit 3 of the Higher Human Biology course. It's looking at neurotransmitters and communication over the synapse, which, if you remember, is the gap between two neurons. Now this is quite a big key area, so I'm only going to be looking at parts A and B, and that's highlighted below. So we're going to be looking at the structure and the function of different parts of the neuron and something called a glial cell. We're going to describe the process of transmission across a synapse and the different things involved there. And then we're also going to consider things called threshold and summation of neurotransmitters. It's all about how you actually change that chemical message that goes across the synapse into an actual electrical impulse in the neighbouring nerve. Here are the SQA notes. There's a little bit of a typo there. It shouldn't say parts A, B and C. It's actually just parts A and B. There's 2 pages worth, so you might want to pause, have a little look. And there's your second page. So there is quite a lot going on. And there's actually, I think, 5 parts of this key area, and we're only looking at the first 2 just now. Right, so first we're going to look at the structure of a neuron and the function of the different bits. So you should recognize the diagram here. It's a typical neuron. There's actually lots of different, well, there's 3 main different groups of neurons, but this is sort of the classic shape of a neuron, and one you'll be most familiar with, the type of ones you see in a textbook. We're going to go over the different bits. We're going to start here at the cell body. So dendrites are the little tentacle-like things that come out of the cell body, and on our dendrites there's actually receptors that receive signals from other cells. And now this is important because when basically a chemical signal comes and binds onto the receptors of the dendrites, if there's enough of a signal, it's going to initiate a response through our neuron. So it'll fire the electrical impulse if there's enough of a signal. Okay, so we've got our dendrites just there, kind of like tentacle things. And then I'm just going to get my pen here. Then moving into this way, here we've got our cell body. Okay, so this cell body basically holds the nucleus, which I'm sure we can all identify by now. It holds the nucleus as well as mitochondria and ribosomes and everything else. Now what's important to note is that if we're starting our signal here at the dendrites, that's where the chemical messages come and bind, then the direction of the message that travels down the neuron is going to go this direction, which is why we are labeling our nerve cell from that direction. So we've got cell body, we've got nucleus, and then what we've got is this long stretch, a long extension of the nerve cell is the axon. And it's the nerve impulse that actually travels down the axon. Okay. Now the axon is surrounded by something called a myelin sheath. And the myelin sheath's main job here is to insulate the axon. So basically it speeds up the impulse that travels down the axon. So once it goes through the axon, it's going to go to our axon or our axon or our synaptic terminals, which are just on the end of these extensions here. And this is basically the furthest end point of our neuron. Now the SQA does not actually specify whether it's an axon terminal or a synaptic terminal. So throughout this course we'll be kind of interchanging between the two, but it's just if you're watching videos or reading a textbook, you know that they're the same thing. So if we follow through with what I was saying about the electrical impulse traveling down the axon and then down towards our axon terminals, this is where it will change into a chemical message and our neurotransmitters will play a role, but we'll get to that bit later. Here's a nice little diagram that you can pause, or if you've got the PowerPoint you can print this off, and it again just covers the different parts of the nerve cell. Now this one looks a little bit different, but essentially we've still got our dendrites, we've still got our cell bodies, we've still got our axons, and here we're calling them synaptic terminals. Okay. Just again, draw your attention to here, the direction of the nerve impulse. It always goes from dendrite to cell body, and then from cell body down the axon. You will never get a nerve impulse going the opposite direction. Okay. Here's another quick slide just to show the three different types of neuron. If we look at our motor neuron down here, this is pretty much the neuron that we're most familiar with in terms of structure, but the sensory neuron is slightly different. It's got its cell body in the middle, and the interneuron has a very short axon. Okay, but the one that we should be most familiar with is the motor neuron. Right, we're going to talk about that myelin sheath right now. So the axon is surrounded by a myelin sheath. The myelin sheath is basically like a fatty tissue, and it insulates the axon. And when we say insulates, we mean that the electrical signal isn't going to get lost anywhere. It keeps it on the axon and firing in the right direction. So the myelin sheath increases the speed of impulse conduction. Remember, a conductor allows electricity to travel through it. Okay. Now the interesting thing about myelin, or the myelin sheath, is we're not born with it. We actually build it up over time. And the actual process of forming the myelin is called myelination. Okay. And myelination is not, as I said, not complete in birth. It actually starts off just in the fetus, and it continues through adolescence, and actually sometimes into your late 20s. Okay, now what I want to do is show you this diagram here. A shows a glial cell, which we will talk about later, starting to produce the myelin sheath. It's starting to encase the myelin around our axon. And then B here actually shows it is almost pretty much the full myelin sheath there. Okay, so it's just a really good visual for you to be able to see what we're talking about when we talk about myelination. Now, just as a side note, myelination, if it starts as a fetus but doesn't really finish till adolescence, and remember that the whole point of the myelin sheath is it allows for fast conduction, so therefore fast reactions, because your impulse will travel faster. This actually explains why babies and toddlers are not really as coordinated or can't react as fast as older people. So I just found this little video of this lovely baby who has been playing with his rattle, accidentally hits himself in the face. However, it takes he or she takes a wee while to actually react. Whereas if you hit yourself in the face, you'd probably go, ow! Probably wouldn't take you a few seconds to realise that's painful. Right, the glial cells, I mentioned them earlier. So this myelination, the process of myelination actually takes place in glial cells. Okay, they produce the myelin sheath, and they also provide support for neurons. Now, if we look at this diagram here, what we've got is, trying to get my little mouse here, this is our neuron that we know very well. And then there's 3 cells around the neuron. We've got 1, 2, and 3. And these are actually different types of glial cells. For the higher human biology course, you don't need to know the different types, but some of the glial cells will produce the myelin sheath, which is what number 2 is doing, whereas other ones provide some structure, which is what number 1 and number 3 are doing, really providing structure. You don't need to know the different glial cells, however, it's just good to know, because a lot of the videos or textbooks that you'll look at later will maybe talk about something called a Schwann cell, and that is the one that produces the myelin sheath. Now, sometimes the myelin sheath can be damaged or destroyed, and there's certain diseases that destroy the myelin sheath, and these diseases often have symptoms associated with loss of coordination or lost ability to speak, and have issues like that. And it's all because if we are damaging the myelin sheath, which ensures that the nerve impulse is traveling down the nerve as quickly and as efficiently as possible, if we lose that myelin sheath, then therefore we're going to alter how the nerves work and how well those impulses travel down. Some of the impulses might be lost. Okay, now I'm just going to get my pen here. Now if you look at this diagram here, we've got an axon with a nice smooth myelin sheath. But then if we look at this diagram here, we've got something called demyelination. And that is when the sheath becomes damaged. And you can actually see the kind of scar tissue and damage of there. So obviously this axon is not going to have an impulse travel down it as effectively, as efficiently as the first axon. We're going to look at multiple sclerosis in greater detail if you're following the tasks on this PowerPoint. We've got a few videos about it that's quite interesting. Now, multiple sclerosis sorry, is a demyelinating disease, which basically means that that axon cover is damaged, and it's mainly in the brain and spinal cord. So if we are damaging the nerve cells with brain and spinal cord, you can imagine that actually has quite a big impact. So this damage will disrupt the parts of the nervous system, resulting in things like double vision or loss of vision. Some people with multiple sclerosis become really, really weak with their muscles, or they can't coordinate their muscles and they tremor, they have involuntary movements, and slurred speech. Now, multiple sclerosis is something that people suffer in varying levels. Some people have to move around in wheelchairs, whereas other people can move around with walking sticks or with frames. It's also something, unfortunately, that gets worse over time. I've put a few videos up in the lesson later, and it will also discuss treatments and how it impacts people's lives. But now we're going to move on from that and just look at neurotransmitters. Okay, so we've looked at the structure and function of our nerve cells. Remember, the electrical impulse is what travels down the nerve cell, but that electrical impulse cannot travel across the synapse, the gap between two neurons. We need chemical messengers, and those chemical messengers are our neurotransmitters. Their job is basically just to take the signal from one neuron to the next, or the next target cell. So it could be that the neurotransmitters are going to a muscle cell or a gland to basically initiate a response in that. We'd normally call them the effector cells. Or, and this is what we'll talk about really through this key area, is that they'll be targeting another neuron. Right. We're going to look at what actually happens with our neurotransmitters at our synapse. Sorry, I've got these little boxes here. They should really come up later. But anyway, what you've got on the left side, yes, the left side, is one neuron sending a message to a second neuron. Okay, and we are zooming in on this synapse here, and that's what this blue box is. Okay. Right, so what happens is the neurotransmitters will actually travel across the synapse. However, SQA likes to change things. It's no longer a synapse. We're going to call it a synaptic cleft. Okay, so the synaptic cleft is the gap between one neuron and then the dendrite of the next, because remember our structures. We've got the axon terminal, or the synaptic terminal, which the impulse has traveled down and then it'll get changed into the neurotransmitter form. And then those neurotransmitters are going to diffuse across to the dendrite of the next neuron. Okay, I'm going to go through that process just now. Right, so first we've got the electrical impulse coming down the axon. Okay. Then, when the electrical impulse comes down, there's loads of vesicles in the axon endings that actually store different neurotransmitters. And we say that they are stored in the presynaptic neuron. Now we're going to be using terms like presynaptic and postsynaptic. So when we're talking about the neuron that is presynaptic, we're talking about the neuron before the synapse. So pre means before. So we've got our neurotransmitters that are in vesicles here, and then that vesicle will move towards the membrane of our synaptic terminal, and then the vesicle will release the neurotransmitters into the synaptic cleft. Okay. So when the impulse arrives, the neurotransmitters are released. Those neurotransmitters will then diffuse across, and remember diffusion, from an area of high concentration to low concentration. So the high concentration is at the presynaptic neuron, and then the low concentration will be at the postsynaptic neuron. Post meaning after. Post is after, so the neuron after the synapse. So we're going to diffuse over the postsynaptic neuron, and it's going to bind to receptors. Sorry, I've lost my wee mouse here. Here it goes. It's going to bind to receptors on the membrane of the postsynaptic neuron. Okay. Now, if enough neurotransmitters bind to these receptors, a response will be initiated. So, and if that response is initiated, so the postsynaptic neuron will receive that message and then do the appropriate response, which could be to fire an impulse, or it might be to not fire an impulse. We'll discuss that later. These neurotransmitters, they now need to be removed, because if the neurotransmitters are not removed, the postsynaptic neuron, the neuron that received the signal, will continuously be stimulated. And look at that, I've got a wee typo here. It says situated. It should say stimulated, okay? So if we don't get rid of these neurotransmitters and they're just floating around, that postsynaptic neuron will continuously be stimulated. There are two ways that the neurotransmitters are removed from the synaptic cleft. One is by enzymes, so enzymes will degrade and break down the neurotransmitters, so therefore stopping that signal. Or the other way is by the form of reuptake. So this is when the presynaptic neuron, the sending neuron, will actually open a channel and it'll take the neurotransmitters back in, and it allows those neurotransmitters to then get reused again in future messages. Now the actual response of the neuron that's receiving the signal depends on the receptors that are actually activated. It's not the neurotransmitter that determines what the signal does, it's actually the receptor that it binds to. So those receptors can either cause an excitatory signal or they can cause an inhibitory signal. Okay? An excitatory signal will result in a signal being passed on through the neuron, so that it will fire off an impulse through the neuron. An inhibitory signal, once the neurotransmitter is bound to that receptor, the signal will not be passed on. So you've got one lot that send a signal, or will try to initiate a signal, and the other one doesn't. So think about it like people that have trained their dogs to speak and be quiet. So train them to bark. Then if they're training them to bark, the command speak or bark would be an excitatory, because it's making the dog go ruff. That was a little bit weird, sorry. Or if their command is quiet or stop talking or something, that would be your inhibitory, because the dog then doesn't do anything. Okay, so we've got one that makes something do something, the excitatory, and we've got the other one that basically makes it do nothing, the inhibitory one. However, it's not quite as simple as some neurons have the excitatory receptors and other ones have inhibitory receptors. Neurons have a mix of both because they have many synapses with lots of other neurons, so they've got lots of different receptors. And basically, what determines whether the signal is going to be an excitatory signal or inhibitory signal, so will an impulse be sent or not, all comes down to the combination of how much excitatory signals or receptors are activated and how much inhibitory signals or receptors are activated. So we say it's the sum of the excitatory and inhibitory signals that determines the overall effect and as to whether those signals actually get passed on. So in this diagram here, we've got some signals arriving with excitatory receptors, and then we've got signals arriving to inhibitory receptors, and basically a signal is only going to pass through this neuron if the strength of the excitatory signals exceeds that of the strength of the inhibitory signals. So talking about strength of signals, there's actually a minimum strength of signals, so a minimum number of neurotransmitter molecules that have to actually attach to receptors to actually make an impulse happen, to actually trigger an impulse. We call this the minimum threshold. So that is the minimum number of neurotransmitters or minimum concentration of neurotransmitters that has to be reached before an impulse is transmitted. Now, if you look at this graph here, we've got voltage up the side, that's our millivolts there, and we've got time along the bottom, okay? Now, this dark solid line here is our resting potential, and this is basically the voltage that is in a neuron at a point where it's doing nothing. When it's just sitting doing nothing, it's still got a certain level of voltage running through it, and that's our resting potential. Now, the neurotransmitters will come and bind the receptors, and that will start, if they're excitatory, it'll start kind of firing off little signals there. Now, if that voltage can reach the threshold, this dotted line, if it reaches the threshold, it will then go and then shoot off right up to the action potential, and that's it basically firing off an impulse. So the minimum threshold must be met for an impulse to actually get fired down through a neuron. If there's not enough of a stimuli, so not enough neurotransmitters, if there's a weak stimuli, so this basically we fail to meet this minimum threshold, then those neurotransmitters are just filtered out by the synapse, okay? They just disappear. And of course, nothing happens, no impulse travels down the postsynaptic neuron. Now, most neurons are not getting signals from just one other neuron, they're getting signals from quite a lot of neurons. So we're going to look at the summation of stimuli. So that is basically the adding together all the neurotransmitters. So a series of weak stimuli can release enough neurotransmitters to reach the threshold to trigger impulse. So maybe the stimuli are coming in little bursts, or they're coming from different directions. Now this is called summation. So basically the sum of the stimuli together reaches the threshold. Now if you have a little look at this graph here, it's got a series of stimuli there. Now this is pretty much like the same that we had. We've got our resting potential line here, and we've got our threshold line there. Okay, so we've got about 6 different short bursts of stimuli here, and you can see it kind of the voltage is going up in a step-like manner every time some stimuli is added in. And it's only after the 6th stimulus that's added in that we actually reach that threshold. And then we fire up here, sorry, but that should be a straight line, fire up, and then we can send our impulse. Now, hopefully, you're all thinking, oh, I remember something about one neuron receiving signals from lots of different neurons. So that would be our summation of stimuli. I hope you are, because which type of neural pathway will lead to the summation of stimuli to the postsynaptic membrane? So this is going back to key area 1 of Unit 3. Drum roll, please. You can do that yourself. It is, of course, the converging neural pathway, or the convergent neural pathway. So remember, you have your one neuron here, and it's receiving inputs from more than one other neuron. And that is basically what we're doing, is we're adding in this input to this input to this input, and then it's going to trigger a response in the final neuron. So that is the summation. We're bringing it all together. So our convergent neural pathways release enough neurotransmitters to reach the threshold. So between the initial neurons, we're releasing enough transmitters to trigger the impulse. And here we've got this little diagram which looks oddly similar to the one before, but it's got different text and different numbers. It's basically saying in neuron 1, it's initiating or is giving a weak stimulus, not enough to cause an impulse to be passed on. Same situation with neuron 2, weak stimulus, but when we add them together, we can then cause the impulse to be passed on. And that is what summation of stimuli is. So here we've got a nice little diagram of summation. So neurons A, B, and C are converging to meet at D, so providing a summation of stimuli in order to get the impulse. Now, if we look here, we've got time along the bottom along this graph, voltage at the side, and the threshold is this level. Now, so neuron A is providing that much of a stimulus. Neuron B has provided that much of a stimulus. Now, remember we said excitatory and inhibitory. So neuron A and B, those neurotransmitters must be getting received by the receptors that will cause the excitatory signal. Whereas neuron C, we're actually decreasing the voltage. So C must be being received by an inhibitory receptor. So that actually brings down our voltage here. However, if we add A and B together, like so, we are actually surpassing the threshold, so we do get our nerve impulse. Okay. That basically concludes parts A and B of Key Area 4. I hope you found this helpful, and again, please ask any questions. And also, like my video if you did find it helpful. It makes me feel good about myself.